Hello, my name is Fatemeh Khalili, and I'll be talking about molecular dynamic simulations of paracellular ion transport. This is the structure of a tight junction, and it might not seem very interesting, but this is the tight junctions are these macromolecular structures that surround the cells in epithelial tissues, and they surround the cells like a belt. And in addition to gluing the cells together, like one belt can be connected to a similar belt on a cell that's next to it, they also control the paracellular transport. So you can see multiple strands here because these structures are usually branched, but here is a simplification of how these structures might look like, and you can see that this would be a belt that surrounds one cell, and this would be the membrane of a cell that's nearby. And when I'm talking about paracellular transport, that would be the transport of ions and small molecules that can actually permeate the paracellular space in epithelial tissues. To give you a few examples, like the best example would be the skin, where the skin is impermeable to nothing can go through the skin because these tight junctions or these pores inside tight junctions basically block the transport of any molecule. On the other hand, we have tissues that are, for example, in kidney or intestines that should be permeable to small molecules. And this is the job of these tight junctions or these paracellular pores in tight junctions. Very recently, in 2015, there was a work published by Christopher Weber that showed that these paracellular pores that exist in tight junctions in fact behave like ion channels, meaning that before people have thought that we have pores that could be open to anything that could pass through. As long as they could fit and they would have favorable interactions with these pores, they would go through. But in this paper, they showed that these pores can, in fact, be gated. So they have states where these pores are open and states where these pores are closed, which was very interesting. In all of these studies, we don't really know much about the structure of these pores. We know, however, that depending on where these pores appear, they can be ions, they can be charge selective, and they can be size selective. So some of these pores are, in fact, selective for small cations, and some are selective for small anions. And at the same time, there is a size limit that's so they would let molecules that are small, relatively small, that are up to maybe 4 angstroms in radius to pass through, but nothing larger than that. And that's the actual current that goes through these pores, but there is also a leak current, which we won't discuss. So the structure of these tight junctions are very complex. There are many proteins that are involved, and you can see here this would be the membrane of one cell, this would be another cell, and there are many proteins that are involved in connecting these two cells together. The proteins that are responsible for controlling the transport are these ones that usually sit, not always, but usually sit on the top of tight junctions and are called claudins. These claudins, this is the structure of one claudin. You can see that it's a tiny, very small protein, actually. It's a membrane protein. It sits in the membrane. It has only 4 transmembrane helices, but it also has this extracellular domain that sits here outside the membrane, and it has to interact with a similar domain, the extracellular domain of another claudin that sits on the other side. So the structure of claudin 15 came out in 2015. However, the structure did not show any kind of pore. And here I can show you the proteins in their crystallographic structure. So we could see an array of protein like this and another one on top. But in this arrangement, you could not see, there was no pore in any form that could be formed if we would repeat this row of claudins above and below and then on each side. However, the structure showed certain interactions between cloudings that we knew were important, and I will come back to that. So it was very interesting, but then we didn't know how the surface of the pore or how the pore looked like, or what was the architecture of tight junctions in general. So the same group who published the structure followed that paper, but another paper that in which they suggested a model of clouding pores. So here we are looking at a strand of molecules that are like a linear strand of molecules that can surround the membrane. And you can see this would be the arrangement of these cloudings in the crystal, but there is no pore. But in this model, they proposed that we should not look for one row of molecules, because there are data that suggest that each strand, each tight junction strand, might actually be formed from it's made from 2 rows of cloudings, which was very interesting, because if you can put 2 rows of cloudings like this, you can see that they form a continuous beta sheet that's very stable. And there was also, of course, cross-linking experiments and FRET data that were also consistent with having 2 rows of clouding in one membrane. That's the top view of these proteins in this model, and this would be a side view. And you can imagine that this one can run continuously. And more interestingly, if we assume that the pores form a structure like this, we would have these half pipes here that potentially could form a pore. So if you have a layer, a strand like this in one membrane, and have something similar to that in the upper membrane upside down, we could potentially have these holes, these pores in there. So that was the suggestion in this paper. So these models are based, this part of the model is based on the data that exists, and they extended that and proposed that there are parts of the protein that we know that interact between the between cloudings in 2 membranes, and they suggested that we can put them together and make clouding pore that looks like this. Of course, this was just a suggestion that this is, if we put these 2 half pipes together, this is how they look like. And more importantly, the parts that are supposed to interact with each other between these 2 rows are not resolved in the crystal structure. And that's because of the crystal packing, because we have them upside down, so they are squished, and they were not resolved in the crystal structure. So what we did was actually we took that suggestion, we built this model, and we modeled these the interactions between these 2 rows. These are mostly disordered regions, and we put them there. We put a lot of effort into modeling these regions so that they could be stable and they could keep these 2 rows together, which was not trivial. We did that part in vacuum, of course, because we didn't want to have the constraints on, for example, having a certain distance between these 2 rows and so on. But eventually, we had a very stable structure. We put it in the membrane. We had a system with two lipid bilayers and then water and everything. And again, we refined these loops and the entire system in a few hundred nanoseconds of simulation. And we could see that, in fact, this model is quite stable. And in this model, we would have these tiny pores that run parallel to the membrane. So in contrast to the other ion channels that transport molecules in and out of the membrane like this, we have these pores that are parallel to the membrane. So the transport is parallel to the membrane, like inside, like orthogonal to the screen. And we have pores that are made mostly of beta sheets, which again exist. That's a form very common in many ion channels. And the size the size of the pore was relatively consistent with what we were expecting. But to verify the model, what we did was we took this system and looked at the ion transport in these systems. So this is just the shape of the pore. You can see that we have a pore that's not circular. It's more like an oval shape. This is the cross sections in each direction. And here we have, for example, the wider region. And I might come back to it if I have enough time. It is not symmetric. So it's like an oval pore. that runs parallel. So these are the two cross sections. So the membranes are up here and here, and these are just if I have a cross section that's parallel to the membrane, and once that's orthogonal to the membrane. So Y would be parallel to. Oh, it's asymmetric. It should be asymmetric. It is not. Yeah, these are the pore radius, and you can see how it looks like. So these are the interactions that we were hoping to be maintained in this model, and we could see that it was consistent with our expectations. But the real verification of the model was that we had no idea of what had to line the surface of the pore. There are experiments here and there that tells us that these are the residues that might face the pore, but really there were very little data on that to map and get a surface. So, but we know that, for example, this clouding has to be cation selective. We took the structure that we had, we applied a voltage bias and looked at the ion permeation. We had to run each of these simulations for quite a while, actually, to get convergence and to have a meaningful data, especially at lower voltages. So for each of them, we ran, I think, about 45 nanoseconds, and you can see that we have a lot of these data points, actually. So here you can see the IV curves for clouding 15 for a solution of sodium chloride. So the sodium current, that would be the blue line, and the chloride current would be the red line, and you can see clearly we have a sodium selective channel, or I have to say a cation selective channel, which was very interesting because we had made no assumption about the surface of the pore or even what part of the protein has to line the conduction pathway. So that was good, and it was consistent with the experiments. But in addition to that, what we did was that we simulated the ionic currents of a few other monovalent cations, which we knew that had to go through the pore. So we had a methylammonium, tetramethylammonium, ethylammonium, and tetraethylammonium. And in all of these, we had cation selectivity, and you can see the magnitude of the current is decreasing as the size of this molecule increases, but this chloride current remains more or less the same. And it was interesting that this was the largest molecule that we could simulate, and it didn't go through. So here we basically had no cations, but still chloride current, which was very interesting by itself because there is experimental data. So these are the data from our collaborators who have calculated the ionic current for clouding 15 so this is they had clouding 15, no clouding 15, and this is the current when there is no clouding. As soon as they introduce clouding into the system, we can see that we have a sodium current or the current for these other cations, and the magnitude of the current decreases with the size of the molecule. Similar to what we have, there are differences. There is actually a difference of 1 point that we can very nicely explain why it's happening. But again, our current was decreasing with size, and so it was very promising in that sense. But what we did next was that we looked at the trajectory of the ions, and we wanted to know why these pores are cation selective. We looked at the residues that interact with the ions that go through. So here we looked at the interaction time. So as the ions pass through the pore, they might interact with the protein, and we looked at how much time they spend near each protein residue. So chloride ions, this is their interaction time, and it seems to be going through very uniformly. So these are the parts, the yellow parts are the parts that actually face the pore, and they are very uniform, and there are parts that just are away from the pore. But for sodium, it was very interesting because it looks like there's a binding site. Sodiums go through, and they spend a lot of time around this residue, which is D55, and two other negatively charged residues that interact strongly with the ion, but this one is very dominant. So what we did afterwards, so this is how it looks like. We have 4 of these residues because we have 4 cloudings in each pore, and they provide actually a very strong binding site for sodium as well as the other cations that we're going through. So what we did next, so this is again iterating the same thing. We looked at the density of the ions. We have these 2 high density regions for sodium, which show the binding site, and you can see 2 of them because there were 4 binding sites, and this is cutting through them. Just you can see 2 of them. So we can see sodiums going through, interacting with the surface. Chloride ions go through the middle, very little interaction with the surface. But more interestingly, we asked our collaborators to mutate this residue to neutralize this negatively charged residue and see how the current is affected. So they could see that, of course, this was the blue is when they had the clouding, they could measure the current, and they neutralized this D55 residue, and they could see that, first of all, the current dropped, and more importantly, this is the relative permeability of sodium to chloride. So that's a measure of the selectivity, the charge selectivity of the channel. So as soon as they neutralized this residue, they lost the selectivity. So you can see that the selectivity, the relative permeability goes to about 1, which was very interesting. And this is what we did. We did this first in the simulation, and then they asked them to do it. So this is exactly what we see in the simulation. We neutralize this residue. This is again the IV curve for sodium and chloride, and we can see we lose the charge selectivity. And we make it positive. We have, of course, anion selectivity. And we did the same thing for a few other residues, a few other mutations, things that we could come up with, and they could also test. And you can see that in a few cases, we could actually reverse the charge selectivity of these pores. So we asked, again, our collaborators to do this, and it was very interesting because they introduced the mutations, and you can see in this graph, it goes up because that's the resistance and not the conductivity. So they did the mutation. They had reduced conductance, and this would be a measure of the selectivity. So that was the wild type, and then these are the mutations in the order how they change the selectivity. And you can see that we can clearly decrease the selectivity of these cloudings by this simple mutation, and again confirming that the model that we have, which we made without any assumption, is actually captures the surface of the pore right. And again, I want to emphasize that before that, we had no idea what lines the surface of the pore or how these cloudings assemble into pores that can continue and make a continuous strand. So in summary, we have been able to actually build a model or even an atomic model of clouding pores in tight junctions that can be extended to a strand that surround the cell, and also and correctly predict the size and charge selectivity of these pores. And actually, we are now able to tell you a little bit more on that, and for example, what goes through and what cannot go through, not because of that particular interaction that I showed you, but because of the way they are arranged. So yeah. And these were not at all possible without blue waters. I didn't emphasize it that much in here, but for all of those points that I showed you in the IV curves, we had to do a lot of calculation that if you wanted to do them one by one, for example, in our own cluster, we could do it, but there were many, many points that, many data that we had to collect in parallel to be able to produce that data. And at the end, I want to acknowledge the students who did the work, Priyanka, Samantha, who actually built the model when we thought it was not possible, and Shadi, who also contributed to the analysis, and our collaborators, Chris Weber and Lee Shen at University of Chicago. And thank you very much.